Hi, and welcome to Nanotalks. This Nanotalk is a recording of our very first liquid phase electron microscopy user webinar that we did with Dr. Jennifer Cookman from the University of Limerick, who talked about her research on pharmaceutical crystal growth using our ocean system. You will now first hear a 2.5-minute introduction of Jennifer, and then her presentation will start. So please enjoy this Nanotalk. And of course, the biggest welcome is to Jennifer. Jennifer did her PhD in 2014 till 2017 at the University College of Dublin in the Center for Bio-Nano Interactions. She focused on synthesis of anisotropic gold nanoparticles for biological applications. She developed a strategy to acquire 3D models of single nanoparticles using electron tomography and use the 3D models to acquire numerical parameters such as surface area and volume, both important for studying nanoparticle cell interactions. Now she started as a postdoc in electron microscopy at the University of Limerick in 2017, working with Professor Ursel Bangert. Let me see, I have a short question. And it's part of the EU-funded FET Open MagnaPharm project to study magnetic field-induced crystallization of organic pharmaceutical crystals. She specializes in studying the crystallization of organic molecular crystals used as active pharmaceutical ingredients with liquid phase electron microscopy. So it's great to have her here and that we can enable her important work with our solutions. I'm gonna now switch over, let me see the presentation and the webcam to Jennifer.
Thanks Olaf for the kind introduction. So today I'm going to talk to you about the experiments that I've been doing with the Lens Solutions Ocean Holder. And the kind of experiments that I've been doing is to study how active pharmaceutical ingredients, which are molecular organic crystals, are crystallized under the electron beam using the liquid phase electron microscopy systems. So first of all, I'll go through the kind of equipment that we have in University of Limerick, and then I will describe to you the main results that I've gotten from the crystallization of an active pharmaceutical ingredient called flufenamic acid using liquid phase electron microscopy. I'll describe to you some of the challenges that I came across achieving these results, and then also how we can relate the observations to existing theories and literature that people have put together in the past. Also some considerations of radiolysis, which is one of the most important things that we have to look at when dealing with liquid phase electron microscopy experiments, and then also the difficulties of acquiring selected area electron diffraction patterns with the ocean holder. So first of all, in University of Limerick, we have a really nice FEI Titan Themis cubed. It's got a monochromator, image corrector, probe corrector. We've got 2 cameras on it, which is great. We have the one view, which allows us to take amazing resolution videos and quite fast videos as well. And then also the Gatan K2, which is a direct electron detector. And for these kind of experiments, it is very important to quantify the amount of electrons hitting the sample, and this is where that detector comes in very handy. We also have the STEMX and also a annular EDX detector rather than the side entry port one. And we have other spectroscopy solutions on it as well. So it's a great instrument, but really what makes that instrument so special is the peripheral equipment that we have surrounding it. So we do have a suite of dense solution holders. So the experiments that I'll describe to you today have been done on their ocean holder, which just allows simple liquid experiments. We also have the stream holder, which allows us to do heating in liquid and also biasing in liquid. We've got the lightning holder as well, which allows us to do heating and biasing in vacuum. And then also the climate holder, where we can do gas experiments with heating also. So it's these kind of holders that really give us the access to do very specialized experiments in native environments and environmental conditions. So as Olaf was describing to you, I'm a postdoctoral researcher in University of Limerick, and the reason why I'm there is to partake in a project called MagnaPharm, which is a FET Open European Commission funded project. So the idea that came about for this project came out of University of Bristol, where they were doing experiments on carnine. So they were doing crystallization of an organic material called carnine in normal conditions, and then also with an induced magnetic field of 1 Tesla. And they found that when they applied the magnetic field to this molecular crystal, they ended up with an unprecedented polymorph, which was the beta polymorph of carnine. Rather than unusual experiments, you would get the gamma form of carnine. These have different unit cell parameters, as you can see on the right. Sorry, I'll just get my laser pointer out. So the unit cell is constructed slightly differently, and then attached to that, it has different optical properties. So the hypothesis of this experiment was then used to apply to active pharmaceutical ingredients that would have some sort of a symmetrical component, like a diamagnetic component to the molecule. And then the project got funded, which is fantastic. So in University of Bristol, they do these experiments at 1 Tesla and 2 Tesla. They do ex situ characterization. We also have Radboud University in the Netherlands, and they do the high magnetic field based experiments, and they can go up to approximately 35 Tesla. We have Sally Price on board from University College London to do the simulations part of the project. And then of course we've got us in the University of Limerick, and we focus on the liquid phase electron microscopy characterization of these crystals and observe the crystal nucleation and growth. And then what we do have to consider in a conventional setup with an electron microscope, because you do have a construct of the electromagnetic lens, you do have automatically an induced magnetic field. So part of my work I do to extract the information about the magnetic field capability is we have a collaboration then in the Ernst Ruska Center in Jülich to do the magnetic field free experiments to then compare what we get in a conventional setup. So this is a schematic of the ocean holder here. So it's quite simple, where you have the silicon nitride sandwich, as I call it, just the 2 windows on top of each other, a system of O-rings, and then it's all shut together with 2 screws. So these O-rings enable the liquid to stay purely within this little flower-shaped bath of the holder, so it doesn't approach the vacuum of the microscope at all. So what I do then is I solvate the flufenamic acid crystals in a solvent, and the solvent I'm using is ethanol, in a range of concentrations so that they're properly dissolved. There are no pre-existing crystals in the solution. The solution is then flowed through the liquid cell holder using a syringe pump, and then we go to the microscope. And we get something like this, where you have the sandwiches, silicon nitride windows, your solvated molecules, and then using the electron beam, then we exploit that energy input from the electron beam to crystallize the crystals of flufenamic acid. So in essence, what you're getting, and in addition, because of the nice detectors that we have, the OneView and the K2, we can get direct real-time visualization of the nanoscale crystallization events by exploiting the electron beam irradiation. And part of that also will be exploiting the radiolysis of the solvent, which I'll explain a little bit later in the presentation. So the reason why I started with flufenamic acid is because it was a nice negative control. So from the results acquired in University of Bristol, we don't expect to see any sort of magnetic field manipulation of the crystal growth to produce an unprecedented polymorph than what we see in normal crystallization events. So I have a frame by frame breakdown here, and the video just on the bottom is playing very slowly. It's a little bit laggy, so I apologize for that. But in the area that's highlighted, you can see that there's absolutely nothing in view. And then over a period of 2 minutes, you do get this nucleation of crystals and dynamic movements of crystals that were already there from a previous experiment, but even the ones that are formed, they cause this like chaotic display of nucleation that have a characteristic hexagonal shape. So our collaborators in UCL did some simulated morphologies using BFTH and also attachment energies to look at the two most stable polymorphs of flufenamic acid and what kind of shapes they would generate. And they found that form 1 would generate this characteristic hexagonal shape, and form 3 would generate these blocky sort of three-dimensional rectangular shapes. So we found in these experiments that we were seeing mostly hexagonal structures, as you can see in the frames in the video as well. We do observe some elongated structures, like I'm pointing out here in the video, but as you will see later on in the video, that some of these elongated structures, they move, and you will see that they're actually the hexagonal shape. And the reason why we see them as elongated structures is on their side they do have a thickness, and this thickness, due to the diffraction contrast, is due to the faceting on the edges of the hexagonal crystal, as you can see is inferred in the simulations. So we found as well, studying the growth of the crystals, that there was an exponential growth pattern, but there were two distinct growth stages as well, an early growth stage and a later growth stage. And it seemed that the hexagonal shape of the particles developed later on. But of course, the magnification that I use is quite large. Maybe if we had better resolution and we could see some more information on the morphology at the earlier stages of the growth. So this work was published earlier this year. There's a reference if you want to read a little bit more about it. Getting to that stage then, when you have your molecule solvated in a solvent, the biggest challenge when doing these experiments is actually highlighting your concentration range. So the concentration I used in those experiments was 50 millimoles. And in classical crystallization on bench for flufenamic acid, the supersaturation is 3 moles. But even at 50 millimoles, when we're using something that's so undersaturated, we're able to achieve a saturation due to the uniqueness of the experiment and the exploitation of the radiolysis products and also the electron beam. So what I'm showing you on the left-hand side is a very high concentration, and I think this is about, I think it was about 150 moles. And on the end of the tubing, because the ethanol can evaporate quite easily, the evaporation led to crystallization of flufenamic acid on the edges of the peak tubing. So this peak tubing is part of the whole liquid cell equipment. So you know then that this can obviously block the tubing to suspend your experiments, or even if this ends up crystallizing between your windows, you just have too high of a concentration, the crystal is too thick, and you won't be able to actually see anything in between your windows. So this has happened, you know, a lot at the very beginning of the experiments. So you know, starting from here, you need to decrease your concentration. So decreasing it a little bit, then you do get a chaotic display of crystallization, and this, you know, it's useful to say, okay, we're getting there, we nearly have the right concentration, but it can't really get much information from this because the crystallization is so fast. The crystals are dynamic as well, and it just happens a little bit too quickly. But then if you go too low, you're literally just looking at an empty window with nothing happening. But then playing around a little bit, you will get to an ideal concentration where you can achieve crystallization of a single crystal, such as what I'm showing on the right here, where initially you may see nothing in the area, but then with continuous irradiation from the electron beam, you will see a nice little crystal starting to develop here. So this is an example of it here. Now you will see that there is a good bit of fluctuation in this crystal, and then in a few seconds you will see that it had created a new nucleation site to generate a new crystal here. And then there's others in view also. So that's one of the biggest challenges when dealing with these organic pharmaceutical crystals, is trying to get that ideal concentration, and then also trying to relate it to the on-batch crystallization, especially when the supersaturation is so high on bench, but then you're using such an undersaturated solution in the liquid phase experiments. One of the things that's related to the concentration then is the observations that we get at different concentrations when you have that ideal range where it's not too high and it's not too low. So we found also with flufenamic acid at the lower end of that range, we were getting these individual particles, and these particles tended to create aggregates, such as what I'm showing you in B here. And with this particular example, and many of the examples that we've seen over a period of time, and in this example is 106 seconds, the aggregate will grow. So it will grow in terms of the number of particles, and it will grow also in size. So we're still doing a little bit of research on this as to how this has happened, or even where these particles are coming from. Yeah, so, and then also in another example here, over the period of 185 seconds, we had an initial aggregate which was measuring at 59 nanometers, growing up to 100 nanometers. So there's a live video of it dynamically moving under the electron beam, and it's growing so subtly you can't really see it when you're watching it, but if you see the first and the last frame, it is quite obvious. With these aggregates that I showed you previously, we did a lovely experiment where we just looked at these aggregates. We just purely watched them for a while in a continuous electron beam irradiation. And we found that there was interparticle interactions between the individual particles, and they transformed into this, we called it a metastable cohesive entity. So this happened over the period of 180 seconds. And then continuously looking at it, we found that there was the transformation between this intermediate into a new phase. And this live video in the middle is showing this what we call densification happening, where in the top right corner of the particle you see this darker mass starting to develop, showing quite a strong diffraction contrast, and the material around it is apparently being consumed to create this new entity into something much larger and much darker. So this act of it consuming the material around it is insinuating that there's some sort of Oswald ripening process happening, where a new energetically favorable entity is being created, and it's consuming the less energetically favorable material around it. Now, the composition of this intermediate is not totally clear. It could be amorphous or poorly crystalline, or a mixture of amorphous or crystalline, but it's energetically interesting anyway that it's able to generate this new phase that appears much darker and has a strong diffraction contrast. When looking at these observations, it's very important to go back to literature and see what people have described and if it resonates with what we see. So what we see here we can describe as non-classical crystallization, and there have been a few very strong theories to describe non-classical crystallization, one of them being crystallization by particle attachment. And this is a really nice paper describing crystallization by particle attachment here. And basically what that is you take these smaller nanoparticles. They can be crystalline, they can be like liquid droplets or purely crystalline. But at the end of the day, what happens is they aggregate and they come together. And one of the actions that this happens by is called oriented attachment. So in the previous example I gave you, we have something that's described like these nanocrystals or these purely crystalline nanoparticles, and they come together to create this new entity, which could be amorphous or crystalline. But the missing information that we have is that we don't see this oriented attachment happening, because usually this would happen through lattice matching. So you would need at least lattice resolution to see the lattices come together, whether it's matching or mismatching. Other theories that we look to is, for example, pre-nucleation cluster theory. And this happens by where you have stable clusters like what we saw before. You have some sort of dynamics happening where they come into aggregates. A liquid intermediate is formed. So this is something like we see that intermediate where the individual particles have come together to form this unstructured mass. And then there's some sort of solidification or crystallization, or what we call densification, to create a final new phase. So, and one of the nice little papers I want to point out on a good description of non-classical crystallization is this one here by Johannin and Helma Colfin. It's a very, very good description to just describe everything to do with non-classical crystallization. Another theory as well is the two-step nucleation, which we commonly see in protein crystallization, where it starts with a liquid intermediate and then you get crystallization happening within that liquid intermediate. So when trying to put all these theories together to describe what we're seeing, we do see that we get possibly pre-nucleation cluster theory throughout the whole process, with some sort of resonance with two-step nucleation theory, because we do have that unstructured intermediate that we don't quite know the composition of, but it's an intermediate phase between the first phase and the final phase that we get. So this energy diagram on the left here was inspired by these theories that where we have a constant energy associated with crystallization, and then you get the final crystal relaxing down to the ground state. Now on the right here is a different take on that, and this is to consider the fact that flufenamic acid is a polymorphic material. It has 9 perceivable polymorphs, and once you have the energy cost of densification, this polymorph fluctuation here is a cascade of where you have your first crystal structure formed, which would be the highest in energy and the least stable, and then it will cascade through the lower energy polymorphs until it settles on the most stable, usually the lowest energy polymorph. Now this has been seen also with calcium carbonate, which I show here. There's a reference if you want to read a little bit more, where you have crystallization beginning with amorphous calcium carbonate and then cascading through the other polymorphs before it settles on the lowest energy calcite. Now there is a part of that that says it doesn't have to exactly go from the highest to the next lowest to the next lowest, etc. It can jump back to other energy polymorphs. It can be quite dynamic before it settles on the lowest energy one. So this is something that we do have to consider with polymorphic materials. It sometimes isn't as simple as just applying a crystallization theory to what we see, but it could actually be an insinuation that the pharmaceutical materials may undergo a quite unique crystallization, which can give us more information on the manufacturing process of the materials. So as I was mentioning previously, with these liquid phase experiments, we have to consider the radiolysis of the solvent. So there's been nice work done on the radiation chemistry of water. This is quite a comprehensive diagram here of how water can undergo irradiation to break up into the different species and further break up to create lots of different products. So in terms of the liquid phase experiments, there's been a few nice studies by Nick Schneider, who studied how water can break up under the electron beam. There's a lot of simulations done in terms of the diameter of the electron beam, what kind of species are generated within the electron beam, how long they stay there, their steady state concentrations, and if they dissipate and perhaps react further outside the electron beam, outside of your viewing area, etc. So we use these kind of studies to base what we see with other solvents. And as I mentioned previously, the solvent that I use for these experiments is ethanol. And we expect it to break up in terms of these two schemes as well. But the kind of species that you get from the breakup of ethanol are in terms of radical species, molecular species, and gaseous species. And the species I want to focus on are the gas-phase species, because these can be both detrimental and advantageous to the experiments, which I'll explain. So this video in the middle is showing you a an example of how the gas products can be almost detrimental to the experiment, where initially at the beginning of the video, which is here, you begin in a liquid phase environment, but as the continuous irradiation is happening from the electron beam at a high dose or flux rate, you do get a lot of the gas species being produced, and what can happen is that gas species can push the liquid out of the cell. And this is what's happening here, where some of the crystals are being carried away from the liquid, and those which have had a strong interaction with the silicon nitride window do remain. So you can see this quite obviously by the contrast difference in the liquid phase and the gas phase environment. One of the reasons that can be quite detrimental to the experiment is obviously it can create a new interface that is uncontrolled, especially when what you're really wanting to focus on is crystallization of your species. And it can also induce degradation of your crystals. So this example that I'm showing here, the electron being dosed was at 32 electrons per angstrom squared. And initially, crystal is quite nice and formed, but then only over the period of 14 seconds, the crystal starts to degrade into pretty much nothing. But before this happens, you do have a very small period of time if you're very careful, and if you use the beam blanker like a trigger all the time, just to make sure you're limiting the electron beam irradiation on the sample. So you are actually able to acquire your selected area electron diffraction pattern in this environment, because initially in liquid, because you are looking through so much material, you're looking through a lot of liquid, including the windows of the silicon nitride. Here's an example of an electron diffraction pattern that I tried to take from this particular crystal, and you can see you're only getting the first order spots. But then if you have this this gas environment, and creating this gas environment isn't always reproducible, but when it happens, it's quite a golden moment, because even just to get that confirmation that your crystals are indeed crystalline, you can acquire this diffraction pattern. So it's quite nice when it happens. So with the ocean holder, one of the things about it, it's a very simple system, it's easy to use, but you are only relying on diffusion of the liquid coming from the inlet through the windows. And for my experiments, I use chips that don't have a defined spacer, so I'm literally relying on the hydration layers between those windows to get the liquid through. This does ensure that the whole construct will be much thinner than, let's say, using a spacer of like a micron or 2 microns, for example. But there have been a lot of challenges associated with knowing if there's liquid between the windows or not. So one of the troubleshooting methods that I use is to condense the beam or the monochromator on an area of the window. And one of the good insinuations to know if there's no liquid there is that you'll just get scarring on the window. Whereas on the other hand, if there is liquid there, you get these black bubbles. Now, the composition of these black bubbles aren't exactly clear, but I know from experience then doing the experiments following this test, we do get crystallization. Now, this is done in just ethanol, and you can get this also in the solution where you have flufenamic acid and ethanol, but you'll get this in simultaneously with the crystallization of the crystals. But this can also happen, let's say if I've introduced the solution of flufenamic acid and I'm not seeing crystallization, it just means that the solution hasn't passed through the windows yet, and it could just be sitting with ethanol. And one of the things, whether you have no liquid in it or you are waiting for the flufenamic acid solution to run through, another troubleshooting method I use is I just tilt the holder to plus or minus 20 degrees, leave it for 20 minutes, go for a run, watch an episode of Netflix, and then come back to it 20 minutes later. And usually that solves the problem, in combination with also increasing the flow rate to ensure that there's more solution running through the system. Another thing that I have been doing as well, it's always a point to note of knowing the thickness of your liquid, even in terms of the resolution that you can achieve. So one of the things I do is called electron transparency mapping, and we use the Gatan K2 detector. This particular experiment now I did in the Ernst Ruska Centre, but I have been able to reproduce it also in University of Limerick as well. And basically what you can do is on each of the corners of the silicon nitride window, just record the dose that's being acquired. So you can see on the right hand side here, we're getting 5.6 or 5.7 electrons per angstrom squared. This means that it's much thicker, and this area would be more appropriate for just playing around with the sample to see what's going to happen, and to just test an area to see, for example, if there's liquid there, if there's solutions there. And then we have an area in the top left corner which has 11.2 electrons per angstrom squared. So that's allowing in a lot more electrons, which means the resolution is just going to be a lot better. So this is ideal to do the final experiment where you make the recording with your optimum resolution. Okay. So I know this was probably a lot shorter than you expected. Maybe that's a good thing. But I hope I have given you information on how we can use liquid phase electron microscopy experiments to study crystallization of pharmaceutical materials. We do have remaining questions on how the supersaturation conditions are created in this very unique environment compared to crystallization experiments that they would do classically on bench. The observations that we see, then it's obviously very important to rationalize what they mean, and also comparing it to literature that's already there and existing crystallization theories. But as I said, for these pharmaceutical materials, because there's resonance of a few different theories, it could be a unique example of how pharmaceutical materials can undergo non-classical crystallization. There's a lot of work to be done on the radiolysis species and how they're interacting. You know, the radical species in particular are highly reactive, but then the molecular species could interact a lot more with the molecules that we're wanting to look at. So there's a lot of work there, and even sort of limiting the interactions of the non-desirable species, such as the gas species. But at the end of the day, I'd say the take-home message is you really need to establish a good understanding of the system being used, whether that be the actual solution that you're using, but also the equipment that you're using as well, just to understand how you're going to get a successful experiment. So next, what I'll be doing is to apply this to other materials, and even in terms of the concentration range. For example, flufenamic acid, as I said, 50 millimoles was ideal for the crystallization experiments, but that was 60 times lower than the classical supersaturation. Whereas for another material, I'm using 150 micromoles. So the concentration range is going to change a lot, and trying to hone in that range that is quite difficult, but we'll get there in the end. So I'll also be working on supporting ex situ characterization, just to further verify the observations that we're seeing in situ in these unique environments. So I just want to say thank you to the PI I'm working with, Professor Ursula Bangert, also Dr. Simon Hall in University of Bristol. He's the coordinator of the MagnaPharm project, so without his grand ideas, I wouldn't be here talking to you. His PhD student, Vicky Hamilton, then she's worked with me quite a lot on these experiments and traveling with me as well to Germany to do other experiments. So she's been absolutely golden in helping me. And then obviously Amir Tababi in the Ernst Ruska Centre, he's been great to help me with the magnetic field free conditions. And as we say, Gaurav Magwiv, and thank you very much for listening, and I'll take any questions you have. Thank you.
Okay, well, thank you very much, Jennifer, again, for a very interesting and I hope also inspiring presentation. So before we go over to the questions of the attendees, I have one question for you, because you mentioned that your institute also has the stream system. So you have both, you have a lot of experience on the ocean system, and you also know the stream system now. So what do you think are the strong points of both systems and applications of these both systems?
I think the ocean holder is a great system to start out with. It's very simple to use, and you know, it's very ideal then for the basic liquid phase experiments, and even, you know, not just the kind of experiments I do where I'm inducing crystallization from a solution, but then also, you know, it's very easy to drop cast materials and do some sort of, you know, reduction experiments with it. The stream, so there's not a lot of control on the thickness of the liquid with the ocean, but the stream does have that control with the kind of syringe pump system. I don't know, maybe Annette could say more about that. But then also the stream does have the capability of doing heating experiments and the biasing experiments. And I think what will come in quite useful for the heating experiments, it would be even as simple as just keeping room temperature, or I see it as quite advantageous for the biological samples as well to keep body temperature constant. So I think that's all I have to say about it. Like, one of the great things that I have experienced with the stream holder is that it's the same holder as the climate holder, which we have as well, and you literally just have to exchange the tip and the tubing. So, you know, for us that was amazing because we already had the holder, we just needed the different tip.
Yeah, so because you're already familiarized with the climate system, it made it also easier to get to know and handle the stream system, of course.
Yeah,
So,
absolutely. You already have the experience there because the way that you put together the tip is very similar to the climate. So there's not too much more learning there.
yeah, you have the full suite, so you have a lot of goodies to work with.
Yeah, we're very lucky.
Very fortunate, yeah. All right, thank you for that answer. Now we go over to the audience. So I'm gonna look up the first questionnaire, and that is Mungai Kamiti from First Materials US. So his mic is now working. Mungai, are you there?
Yes, can you hear me?
We can hear you, yeah. Welcome.
Yes. The question I asked, this is very interesting, and thank you very much for such a very nice presentation, Jennifer. Voila. Now, the question is, why do you think you formed those non-crystalline coagulation, and what is the driving force?
Yeah, it's not very clear because they're so we believe it's driven a lot by the radiolysis products and how those radical products interact with the flufenamic acid molecules. So on the flufenamic acid molecule, there's a carboxylic acid functional group on that, which can create a resonant kind of carboxylate structure anion. So I believe that plays with the energetics quite importantly to create the unit cell, to construct the molecules into the unit cell structure. But we need to do a lot of more work to actually properly quantify how that happens, because it does seem like a bit of a mystery at the moment. But yeah, there's a lot more work to be done, but we have theories that we're working on.
As you might have guessed, I am a colloid chemist, and particle interactions is my bread and butter. So when I see when I see particles going like that, there has to be a number of forces in place, and that's why my interest. I know crystallization is when they are falling out of solution. Nature forces them to be crystalline or amorphous, but some of them were like chain-like, and that's very interesting because we need to know how particles come together. Is it because they have their own van der Waals forces or electronic forces, and those some of them are long range and some of them are short range? So this is what I do. I study particle interactions of the same dimensions you are looking at. That's why I put the question there, because I would like to know why this anomalous behavior. It should all crystallize. It should not be forming those chain-like that look like fumed silica, like necklaces.
I understand, and there's one additional thing that I didn't mention in my presentation, but it is known as well that the electron beam can cause nanoparticles to be attracted to the electron beam. So there has been a number of studies on small and large nanoparticles where they would be completely in a different area, but when you condense the electron beam, then they will be attracted to it in a liquid environment. So that's another contributing factor that we'd have to consider then as well.
Thank you.
All right, thank you very much for your question and the nice discussion. So the next one in line, if I say it correctly, Seung-Young Lee from Lawrence Berkeley National Laboratory from the US. So your microphone should be working now. Are you there?
Yeah, thank you very much.
Yeah,
thank
welcome.
you for the nice presentation. Could you please explain more details about like how you obtained the electron transparency mapping for measuring the thickness of the silicon nitride membranes?
Yeah, so what we do is being really technical about it. So you have your holder in the microscope and you record what is being detected by the detector, and then we take it out without changing the electron beam at all and measure that as well. So essentially what should be hitting the top window of the holder will be what is hitting the detector without the holder in it. So is that right?
So just in a like TEM mode, and you change the positions, right?
Exactly, yeah, that's it. It's as simple as that. And it's literally just to get a measure of how well the electrons can transmit through the sample. So it doesn't give a quantifiable measure of the thickness per se, but it does give you an idea of the ideal regions of the windows to do the experiment in.
Okay, thank you. And I have another question. So you said that the tilting can help to diffuse the liquid, but what do you think of that principle?
Yeah, it just like I don't know. It seems to work in combination with leaving it for a while and then also increasing the flow. So I just, when I have that problem, I sort of hit everything at it. And it was a troubleshooting method also that was suggested by Dens as well. So I had some sort of support there to be like, okay, tilting might actually work, but it's hard to know exactly which side of it is the inlet or which side of it is the outlet, because the tubing can twist inside the holder as well. So there's that kind of complication as well. So I usually do a bit of both, where I do maybe 15 minutes at the plus 20 side and 15 minutes at the minus 20 side, and just see how we get on.
Okay, thank you very much.
Okay, thanks.
And that brings us to the end of this Nanotalk. To stay informed about upcoming webinars, I invite you to follow our social media channels, LinkedIn and Twitter. I also invite you to sign up for our monthly newsletter, in which we share interesting in situ research and technological innovations. I want to thank you for watching and hope to meet you at one of our next webinars.